And if you could follow what I was asking. Okay, so the first step in getting from DNA to proteins is called, say it like you own it.
Transcription.
Called transcription. That step happens in the nucleus, and the end product of that is going to be what? mRNA. If you said RNA, I'll give it to you, but you know it's a special kind of RNA called messenger RNA. Is everybody good with that? Second question. The second step is called translation, and it happens in/on. And if you said ribosomes, great. If you said in the cytoplasm, out in the ER, that's great. But you know there are workbenches called. So if you said those things, that's great. And the end product of that step is that you translated the messenger RNA into a what? Protein. So did everybody follow that? Okay, great. The law of complementary base pairing, I wanted you to explain it, and you just had to be brief. It states that only certain bases can pair with other bases, and you do need to know that adenine pairs with, unless you're talking about RNA, and then it's not thymine, it's a, and cytosine pairs with, great. But if you said for this quiz that it's the understanding that it's very specific in how bases will pair, that's fine. You will need to know that A to T, C to G, T or U, depending on DNA versus RNA, okay? All right. Quick test. How many total chromosomes do humans normally have? Total. 46. If I'd asked you for pairs, you would have answered it. Okay, right. List the 3 types of RNA discussed in class, and you just had to put messenger RNA, transfer RNA, and ribosomal RNA, right? Little m, little t, little r, right? What is a codon? What is a codon? Who put an answer to that? It's a set of 3 nitrogen base pairs. It's going to be coding for something, right? But it is. It's a set of 3 nitrogen base pairs. It's a set of 3 bases. And how many different codons, how many different sequences of 3 do we have? 64. And you all tell me, I didn't ask it for here, but what do we know about the 64? We know exactly which amino acid each one of those 64 calls up for that site. And what is that knowing?
The primary structure.
That's knowing the what? The genetic code. You should understand that for this test that's going to be on this material. Now, of course, this isn't on the test you're going to have Tuesday, is it? But for the next test, you have to have a thorough understanding of this field to answer multiple choice questions about it. So hopefully, go back and review it if you didn't get it from here. These quizzes are to let you know how well you're staying up with it, okay? So fun. Define mutation. Who wants to tell me what they put?
A change in the DNA sense strand.
It's a change in one of the bases in that DNA sense strand. That's exactly right. That's what it is. It means one of the bases have changed, sequencing. And that means that the message would get potentially changed. And the codons might be different, which means that it might end up having a different what in the chain? A different amino acid than where it should be, right? Okay. I'm talking loud, like, you know, when people are speaking a foreign language, if you talk louder, they'll understand your foreign language. This is like so not true. But did you get it? Or y'all, do you have any questions? All right. No, one question.
We were discussing this before. I had a definition that it's a benign change at this level, but mutations can be good or bad, right?
You had written down that it's benign?
Yeah.
Okay, so that's when you would have wanted to see, either by listening to recording, this is such a great comment here, because you would want to see by listening to recordings or checking other people's study guides. I said it could be benign, but I told you that they can also be, it can be devastating, or they could be good. They could be an adaptive. So mutations can be any of those 3. The ones we tend to hear about are the what? The devastating ones, right? But it could be any of those things. It could just lead to a variation in the trait. that, you know, like blue eyes. You know, it could just lead to variations in traits like moths being white or black. That was a mutation that occurred way back down the line. They even know when it happened, you know? So that's just a mutation that happened on the DNA sense strand that changed the color of the moths, right? And that mutation ended up being an adaptive mutation that helped some of those moths to survive because the environment was changing. I don't know if y'all remember that little exercise in your general biology class where you did the peppered moth experiments, but that's what that was about. And that's happening all the time. It's happening to our microbes that are in our system, that are in our bodies. Those mutations are happening, changes are happening, and we know that. That we understand that mutation happens at a particular rate in every living organism scientists have studied supports the theory of what? Of what? Because evolution simply states that the percentage of traits found within a population will change over time, and that we know mutations occur at a spontaneous rate. That supports the theory of evolution. Right? So, okay. That should be sort of intuitive. Okay. Define mitosis. What you put? Cell division. And then I wanted you to tell me a function of it on the next question. So what you put? It, for development, for growth, for regeneration, for repair. All of those things are why we need mitosis, cell division, which is what kind of cell division? Going from one cell to two new cells. The one cell, when you're talking about mitosis, I didn't tell you all this, but that first cell that's dividing is called the parent cell. And then that parent cell makes a copy of its DNA, and now we've got two new cells. Y'all remember that with mitosis? The two new cells are called daughter cells of the parent cells. And are those daughter cells different from the parent cell? No, they're what? They're exactly the same. And that's a type of what? Cloning. So the mitosis that's going on in our own cells is a type of cloning that happens in cell division. So mitosis is a type of cloning because the two new daughter cells are identical to the first, the parent cell. Okay, that was way more information. All right, so that's the function. Lots of functions: growth, regeneration, repair, development. Right? What are two main phases of the life cycle of a cell? What was the phase that a cell spends its most time in? Interphase. And what was the phase called where it's dividing? The mitotic or mitosis phase or mitotic phase, right? All right, so good for you. And then what the bonus was, what does being heterozygous for a trait mean exactly? What does that mean that you're heterozygous for a trait?
Having you carry both the dominant and recessive traits.
Then somebody said different. You said that you're going to have, from one parent, you will have inherited one set of information, from the other parent, different information. And we think of them as dominant and recessive, right? You're hetero. You've got different information for that trait. This is all important information, language that you need to know going forward, because when we start talking about reproduction and sexually reproducing organisms and what's happening in your gonads, if you don't have the basis of this, you might be a little lost. So the reason we, you know, I really encourage you guys, and I hope it's seeming like encouragement instead of like, I don't know, fussing at you or yelling. I hope it seemed like it's because I know, I was in clinical medicine for 15 years. I know that the people who come that have the great foundations, they're the critical thinkers. They're the best practitioners. And you know what I call that? I call that having common sense. Because we sometimes try to jump to the next level without thoroughly understanding it. And then you know what? A lot of people can do it because they have photographic memories and they're great at memorizing, and is memorizing learning? No. I've seen it repeatedly. It's not. You want that person with you that actually has the common sense, meaning that they have learned it from the foundation up, right? The foundation up. So I'm presenting this to you in a way that I'm breaking it down, and you need to own it as we go. Because if you don't, you're going to struggle. Your struggle is going to be continuous, right? And you guys, I could just not care and be reading the PowerPoints, but I do care. I want you all to get it, okay? I want you all to get it. Do you have any questions for me? Were these fair questions for this lecture that we just had? All right, good. Let me tell you before we continue on with chapter 4, because you all know chapter 4, is not going to be on the test, Susie. And I absolutely understand that you all are busy, so you probably aren't going to be looking at chapter 4's information. You probably aren't going to be looking at this until after Tuesday. I get that, because I would want you to spend your next few days reviewing the insights. Everybody know what a deeper insight is? In chapters 1 through 3, reviewing those, reviewing your lecture notes, going back and looking at those lecture study guides, you know, looking at those other people's lecture study guides. Some of you are not putting in any time on those study guides. And do you think that nobody's noticing that? You know, because some of you are getting things wrong on them, all right? And it's just real obvious. And others of you are really doing a lot on the study guides. Guess what? I can tell. There's a direct correlation for people who are doing their study guides well to how the lecture quiz grades are going. Isn't that amazing? Okay, that was sarcasm. But you know, it's true. It's for you. Those study guides are for you, not just so you're checking off boxes. They're for you. And then using other people's study guides, even if your study guides are great, using other people's to check yours, see how complete you're, that's helping you to learn. So use that resource, right? Some of you don't need them. Some of you don't need the lecture recordings. Some of you don't need them. Some of you are audio learners, and you know that if you hear it, you'll own it. I have a child that's like that. And he taught himself to be like that because he had to read some whatever. But he is an audio learner, and if he hears it, he never forgets it. I had a student one time, his name was Franklin. I'll never forget him. He sat in class, and he never wrote a note, but he listened. He was like so intense, you know, listening to me. It was like the third day of class, and I was like, Franklin, you're doing fine on the first lecture quiz, but why aren't you taking any notes? And because he was like bothering me, the way it was like so focused in. And he said, because I have to hear it. I have to hear it. I'm an audio learner. It's the only way I can do it. So I get that we're all different, but figure out soon what you need to make it work, okay? And y'all are just starting, so don't want anybody to be too nervous about it, but there are a lot of resources for you in this class. So you have tutors, you have lecture recordings, you have study guides. A lot of tutors, I don't want you to feel anxious about this first test. Those questions will be just like questions from here, where when we go over that test after you take it Tuesday morning, I'm going to say, were any of those questions not in your lecture notes or not in the insights? And if you can tell me that there is one, guess what I'm going to do to the question? Throw it out. So feel comfortable, feel assured that you know what to study. Are you going to memorize PowerPoints? No. Bless your heart if you're trying to do that. Okay, don't do that. But you know what to study, and I want people to feel comfortable about that, because that is going to be the truth. One more quick thing, the lab quiz, that microscope. Let me say it again in case you didn't have it, in case you missed the lab, or in case you weren't, whatever. That lab quiz will be at the beginning, so you'll do that. It'll take probably 2 minutes. You're going to be labeling the microscope, and I only gave you certain things to label because the others are just busy work. But here are the things you need to label. The oculars. If you want to take a second to get out your microscope picture that you had, then that's fine. Yeah. Did anybody else miss the lab? Anybody else miss the lab? All right, because I have, okay. So the ocular lenses, I'm going to have you label. The objective lenses, the stage, the condenser, the base of the scope, and the light. and the coarse adjustment knob, which is the big one, and the fine adjustment knob, which is the what? Do you see that there's two lines going to those adjustment knobs? One is the big one, that's the coarse, and then one's going to the middle, which is the little fine one, right? The little knob. Hey guys, that's it. Is everybody good about that? You got the list? Is it going to be anything other than that? No. All right, now listen up to this, because these two questions would be fair questions to ask. The first question I asked you in lab was, or we talked about, and I gave you the answer, but here it is again. So you're just going to have to vomit this back to me, okay? And that's okay, because you're going to get a 100 on this, okay? So the first question I asked you was, how do you determine, when you're looking at a specimen, you're looking through the microscope, how do you determine what the magnification of that was, that specimen you're looking at? How do you determine that? Who's got the answer?
The ocular magnification times the objective lens magnification.
Did y'all like that? Did y'all agree with that, or did y'all have something different? The ocular lens magnification multiplied by whatever objective lens magnification you're using. Why couldn't I, Ashley, since Ashley's vocal this morning and I appreciate her help. No, I appreciate Ashley helping me, because, you know, sometimes,
That's on.
I mean, Andrea. I don't know why I'm saying Ashley. Ashley with an I. I don't know why I'm saying that. Andrea, thank you. But Andrea, why didn't I tell people you could answer it by saying 10 times 10 or 10 times 40 or 10 times 100?
Well, the ocular lens can change. It can be 10.
In your clinic, it might end up being 12 or 15. You got to look at it, and it'll be on it what it is. And then you might have different objectives than we do too. So that's why that wouldn't be right. And if you write that, you're going to get it wrong. It's going to be counted points. So don't get that wrong. Do exactly what Andrea said. All right, second question was, tell me why it's important that you're always keeping in mind what the magnification is, so you can identify what you're seeing. So for identification, and what we said was that some things look exactly alike, but what's the only thing different about them? Their size. So you've got to know your magnification, because they're going to help you identify things. It's going to help you to identify. I have had, and I don't think I got to tell you all this is true story. I had a resident. This resident had gotten through undergrad. I don't know what he had as an undergrad degree. Gotten through med school, gotten through internship. He was a resident, came in with a slide from a patient, was so excited because he thought his patient had this fungal infection with a fungus. But he wasn't looking at his magnification. It wasn't a fungus. It was not only that patients didn't have a fungal disease, which means they didn't need to have systemic antifungals, which can kill you, by the way. The medicine can kill you. This patient didn't even have a bacterial infection, because the bacteria he was seeing, which are much tinier than fungi, if he just known what magnification for that, because the bacteria he was seeing was part of their normal flora. Do you get this? You've got to know, be consciously thinking about how many times about what objective am I on? What's my ocular? What's my magnification? Is this really what I'm thinking it is? What's the size of this thing? Right? True story. So please, you know what? That's one of those people who were down here and they jumped up. Do you get what I'm saying? And could do it because, you know, smart in some ways, right? We're all in it together. It's a team. So we're in it together, and I want you to do well. Are you going to get a 100 on that?
Yes.
Thank you. I am also going to give you all an opportunity today in lab to, do you remember this, the quiz you just got back, this quiz where you had some of the structures that we talked about. I don't know if it's all of them, but it was a lot of them. I can look right now, though, and see it wasn't all of them, but some of the structures that match their function. Today in lab, I'm going to give you all an opportunity to color. And what you're going to do is you're going to use this, because some of you didn't have it the other day, but you should have it now. after the lab today. You're going to use this, and I'll give you instructions, but you're going to be doing something that's going to count as a lab quiz participation grade on cytology. What is cytology? Study of cells. Cells, that's right, study of cells. So you're going to use this, and this is going to be just everybody doing it and getting checked off what they did. A 100 lab quiz participation. Can we say hallelujah? Okay, so you'll have a few of those things throughout the semester. But what this should do, you think I'm doing this just to give you a 100 on a lab? I'm doing this today so that is this going to help you with the multiple choice questions, answering these questions? Some of you got them, so you already know them. Thank you. Helps you with your peers today. So you got them, but others didn't. So it'll just be reinforcing what you'll have on the test Monday. So that'll be a participation thing to do in lab today. It won't take too long, but I do want you all to do it, and I'll tell you more about it when we get there. Okay, so we good? Ready to go? All right, so we're talking about inheritance, aren't we? And we said that there was some misinformation about genetics, just like there was for the theory of evolution. Misinformation, misunderstanding, whatever. But here's the thing, we don't want to spread misinformation, do we? So we want to make sure that we understand what's going on. And am I in the right chapter? Okay, yeah, I am. So we said we get this idea that, we know what cytokinesis is, by the way? It's a doubling of the cytoplasm so that the two new daughter cells are going to end up having all the supportive structures they need when they're formed. So that's exactly right. Everybody remember what a karyotyping is? It's a physical analysis. We're physically looking at the chromosomes, and they can tell us something. Yeah, and so genetic counselors especially, because they can tell me only a few things because I don't really work with this. And I know a few things. We're going to do karyotyping in the lab today as well. So you guys are going to see a little bit more about this and understand this. But a genetic counselor would be able to look at one of these and know dozens of different chromosomal anomalies that can happen. Can a karyotyping tell you everything? That can it assure you of everything? No, it can't. But it can tell you. It can tell a genetic counselor a lot just by doing that. So now we're talking about inheritance, and here's what we were talking about. We were talking about the idea that dominance, a dominant trait means simply that you have information for a particular trait to be expressed. And a different word for trait would have been gene, characteristic, or recipe for a protein, right? So that's what we're talking about, right? And if you have the information, do you have to have it from both parents or both homologs? Or can you just have it on one? Just one. And it will be expressed, right? Okay. So recessive, we were saying, it means that it's not that if you don't have that information and or you could have misinformation, right? So we said some misinformation out there is that dominant traits predominate in the population, and they don't. They don't. So that's not true either. It's simply at a molecular level an understanding of this, right? Okay. So actually, before I move to something a little bit different, I want to come back to this because it is true. And you're not going to look at this until after Tuesday. I get that. But I would hope in your notes you would write some of these things down, some of these things we practice, because you will, on the test that has chapter 4, you will have to solve some genetic problems, right? And I will tell you, I'll give you some examples. You ready? So I will tell you something. I think I gave you a couple the other day, but I will tell you the genotype and or the phenotype of parents, and you will have to do Punnett squares to answer questions about the probable outcomes of offspring. Are you good with this? Okay, so we did one the other day, I think, maybe a couple, but we had to know the language and what we're talking about. Phenotype means what you can see. So you can either see that someone's dominant or that they are recessive. Genotype means that you know both homologs, what they're holding. Genotype means you know the information on both homologs for that trait. So if somebody is recessive as a phenotype, do you automatically know their genotype? Yes. Because if they are recessive, could they possibly be, and I'll just use the letter A. Could I use any other letters? Be fine, but I like to, we'll just use the letter A. Right? Could they possibly be holding a dominant? No, because that wasn't the definition of what dominant was. If dominant was there, it would be being expressed, right? So if somebody has the phenotype of recessive, you know their genotype. You must understand this, okay? But if somebody is dominant as a phenotype, you don't necessarily know their genotype because they may either be homozygous dominant or they might be heterozygous for that trait. They would still be phenotypically dominant. Do you get that? Okay, so let me say this. For the questions that I give you, I will always give you enough information to answer the question. So don't get like blown up, you know, just take a breath and read the question, all right? So if I were to tell you that one of the parents was phenotypically recessive, do you know how to write their genotype? Yeah, how do you write it? Lowercase, lowercase. And if I were to tell you that the other parent was heterozygous, do you know how to write their genotype? Okay, is everybody good with that? All right. And what did we say that X meant? Maybe. So, and is this for each time they mate? These are the probabilities we said. So this is kind of what we were leaving off. But I told you to do a Punnett square for each trait. For one trait, there will be a potential because of sexually reproducing organisms. For each one trait, there will be the potential of 4 different outcomes for the offspring genotypically. And I said it's easy kind of to do this because all you have to think about is what the parents could offer, right? So if this is the mom, do moms get to send both of their homologs to an egg, or do they only get to send one of each pair? One of each pair. So we said the mom could either send to the egg, this one, or the mom could send what? And the mom could, and the father could send 4 sperm, could send, or what? So if I were to ask you if this egg met that sperm, what would this child be? So this child is what? This child is phenotypically dominant and genotypically what? Genotypically heterozygous. What about this child? This child is phenotypically dominant and heterozygous genotype, right? What is this child? What is this child? So if I had given you this information and told you that it told you it was an autosomal recessive disorder. Autosomal meant which chromosomes were involved. 1 through 22. The first through the 22nd pair. And it's a recessive disorder, meaning that they had to be recessive. They had to be homozygous recessive. They had to be. If I were to tell you that a mother who was heterozygous for the trait, did not have the disorder, made it with a father who had the disorder, autosomal recessive disorder, then what's the chance for each time they mate that their child would have the disease? What would you say? 50%. Why would you say 50%? Because this child has it. And does this child have it? Yes. That's 2 out of 4. That's 50%. If I were to ask you what is the probability of a child being a carrier for that, now pay attention, be careful. If I was, what'd you say? Why do you say 100%? Because these certainly carry the information, but these do too. They will, right? We tend to think of carriers as being silent carriers, but yeah, I think it's more accurate to say 100%, right? Any questions about how I just did that one? All right, so here's what, I think I've done a couple. So do y'all need any more of those? Do you feel pretty good about those? You do? Okay. Just move on. Okay. All right. Okay. Here's what I want to let you know too. I want you to write down the name Gregor Mendel. Gregor Mendel. And I want you to write down that he was an Austrian monk. And then I want you to know a little bit about him. He lived in the 1800s. Did we know in the 1800s that the DNA molecule carried the information for our traits? No. No, we didn't know until well into the 1900s. We thought, before then, molecular scientists thought that proteins were inherited. They didn't realize that proteins have a shelf life and die pretty quickly. Get denatured and whatever. You don't inherit that. The DNA molecule that holds all the recipes for what proteins can be made. But we didn't know that until into the 1900s. Mendel didn't know that. Right? Did Mendel have the electron microscopes to be able to see inside his cell? No, he was an Austrian monk. And so he lived in, really kind of almost isolation a little bit, but not really. But he was also incredibly inquisitive. And his observational skills, they were phenomenal, his observational skills. And what's the first step of the scientific method? Observation. And from observation, you will ask a question. And from the question, you will answer it. That's a hypothesis. Then you test, and then you figure out your results, right? Well, not only was he amazing in his observational skills, and his questions that he was asking about things, because it was thought that this was what was thought in the 1800s, that people inherited from parents, or, he wasn't just looking at people. People were probably the least of his worries. He was looking at nature, really. But he said, traits don't blend. Traits, and he said, traits don't blend. Y'all might be thinking, yes, they do, because I look half like my mom and I look half like my dad. No, they don't. And I'll tell you, Gregor Mendel was right in that observation. They don't blend. Traits hold true, and he told us that. Gregor Mendel is the father of modern day genetics. Every single one of his theories are holding true at a molecular level today. Is that crazy? Let me tell you why that is. It's because of his, you could say obsessive. You could, maybe. Obsessive, meticulous inquiry. And the results he got, the numbers spoke to him. And he didn't allow them to lie to him, to himself. They actually spoke to him. He did so much testing, and he had so many results that the number of the results he got, he was understanding what those numbers were saying. And those numbers were saying to him that traits don't blend. That is a myth. It is false. And actually on a large scale, it makes sense to us, because if traits blended, wouldn't eventually we have just a homologous kind of soup? Wouldn't we all kind of be ending up, everybody looking alike, like they did? But that doesn't happen, because he's right, and he's right at a molecular level. This whole little Punnett square I did for you, and thinking about traits, he gave us that. Not understanding that it was a DNA. He didn't need to. I mean, it's really amazing. Seriously, amazing what he did. And so he had so many different interests, but he was phenomenal. And so I really want y'all to know that. He also said that in a species, because we've just been looking at one trait at a time, right? And how they are passed. He said that traits get assorted independently. It's called independent assortment, the theory of independent assortment. And guess what? That's true too. That's true too. And he even talked about, with certain species, what kind of probability that that you had for the mixing of those traits. And those numbers are true too. So I want you to think about that. I want you to think about what you could be doing if you weren't on your phone or watching TV. Because you didn't have either. There would be a lot of time. Think about that. Maybe why people who had the ability to do this, that people used to speak like 5 and 6 languages, right? Had time for that kind of stuff without the screen time. I'm going to think about it. Okay, but anyway, I'm just as guilty of the screen time. Okay, it's just amazing. People are amazing. I want you to here's one thing, and again, these numbers have held out true for this. I want you to think about this, because we just talked about homologous chromosomes. So the first through the 22nd and the 23rd pair, there's pairs. And we said for men to produce sperm, they can only send one from each of the pairs to the sperm. Is that right? They can't send 2, or they shouldn't. Okay. And women do the same. When women make an egg, you can only send 1 from your pairs. Do I need to go back to the karyotyping for you to visualize that? But I want to, because it's kind of important that you get that independent assortment. Mendel told us this. He told us that when you're thinking about how traits are inherited, it's not like the mom's going to send this one, the first one to one egg, and then the second pair to the next egg. That doesn't happen. He said you might send this one this time, and then this one, and the next one would be completely shuffled. And he said the combination would end up being, and this is what I'm going to tell you. I want you to think, because this is, and don't write this down, I just want you to imagine this. Men start producing, women don't have as many eggs as men produce sperm. So we'll talk about men here. And this is true for women too, but it's just not as impressive. But women have hundreds of thousands of eggs. And each month, once cycles start, usually it's just 1 egg a month, unless a woman inherited, they could do 2. But anyway, it's usually 1 month. All right. So in women, it's more about quality than quantity. We think about, but I want you to think about with men. That should be quality too, ladies. Okay, but anyway, but I want you to think about men. Men start producing sperm at puberty. That's actually what's called puberty, the first viable sperm, and then ejaculation. And they're at puberty. And men can produce viable living sperm if they live to be 110. All right? Every day they're on a cycle of making sperm, right? They're on a 24-hour cycle where women have 28-day cycles, right? So I want you to think about sperm and how many sperm a man who lives to be, let's say, average age of men, 76, 78, whatever average. How many sperm they'll make from puberty to that age on a 24-hour cycle. And to give you an idea, let me tell you this. In a teaspoon of semen, because everybody can imagine what a teaspoon looks like of seminal fluid. And it's got 5 cc's or 5 mLs in a teaspoon, right? If you didn't know that, you know it now. There's 5 mLs in a teaspoon. You don't have to write that down. I'm just telling you to picture this. 1 ml, and there are 5 of them in a teaspoon. 1 ml can have 150 million sperm. So I want you to get your head around every teaspoon of ejaculation a man will have in his life, and he could have 150 million sperm times 5 in each teaspoon in his whole life. Now I want you to hear what Mendel said, and he's right. No 2 sperm a man will ever produce in his lifetime will be exactly alike. That's the kind of assortment we're talking about. And there were 2 things, not just independent assortment, but something called crossing over that happens. And Mendel was right. That's exactly what happens. He didn't know about karyotyping, did not know about nucleic acids, did not know that, but he knew that's how traits would be inherited. By looking at just a few traits on the species that he cross-bred, and the numbers, what they told him, and the results, he came up with those theories, and he's absolutely, like, I had chill bumps. Do y'all? Think about that no two sperm a man will make in his entire life will be exactly alike as far as genetics goes. That's like mind-blowing. If it didn't blow your mind up, you weren't listening. You stopped listening to me, and it was my storytelling that was at fault, not y'all. Okay, so there you go. Let's do this. I want you to know, and I want you to answer. If I ever ask you, do traits blend, what would you say?
No.
No. You're going to remember Gregor Mendel, right? An Austrian monk who did. Y'all should go back and read about it, because it's pretty amazing, all the stuff he did. So, okay. I want you to know, though, you'll see things that can fool you, because what you'll see sometimes, and let's, instead of humans, let's think about, well, you can think about humans, but let's think about a plant. If you had a red flowering plant and a white one, and you cross-pollinated them, pollen is sperm, is plant sperm, and you cross-pollinated them, you would get pink as the 100% pink in the first generation. Now, wait a minute. That sounds like blending to me. Does it sound like blending to you for that trait? That sounds like blending, doesn't it? But Gregor Mendel didn't stop there. What he did was he took that first generation of 100% pink and he cross-pollinated them. And guess what he got in the next generation? He got a lot of pink, but he got some white and he got some red too. Which meant, and which told him, which spoke to him, that the trait for those colors were holding true. They were just being masked, right? That they were still there. They were still there. They had not changed, but they were being masked, not blending, not blending. And that's how you understood the heterozygous condition versus the recessive and dominant. You get what I'm saying? Same thing in people, we can kind of see with skin tone, where you'll see skin tone, several generations, then all of a sudden, here's this next generation that looks really different, right? It kind of looks different. So traits can sometimes be hidden and masked by other things alleles that can influence them, and then you see them because they were there the whole time. They did not blend, they did not change. At a molecular level, they held true. You hear what I'm saying? That's traits don't blend. All right, now I want to tell you about something else that you know about, though. For some traits, there's something called co-dominance, which means that one won't mask the other. We said if somebody's heterozygous, their dominant is masking that they're holding a recessive, right? Like, did I know I was holding a recessive until I had a child that was recessive? I didn't know that, did I? I didn't know that I was holding a recessive. I really didn't. Because both my parents were dominant too. I didn't know I was holding recessive. So until I had a child that was. That's dominant recessive. But there's some traits for some disorders, and we tend to think about disorders. There's some traits that are co-dominant. And the best one to give you an example of, and this is one the book does, is blood typing. So I'm just, again, not that y'all care about me, but I'm a good one to use for this. How many of you know your blood type? Oh, a lot of you. And Amy too, you do your blood typing in here if you want to do your blood typing. But anyway, so I know that I have the blood type A, which means this is ABO blood typing. We'll talk more about it when we get to AMP2, and we're in hematology. We'll talk more about it. I know I have the blood type A. And did I tell y'all I made it with somebody? Yeah. All right. And guess what? I didn't ask him his blood type. I didn't care. So I didn't care about his blood type. But here's what we did. And again, y'all took my word that he's the father of my 3 children. He really is, because I would tell you if he wasn't. But he is. So anyway, I found out, though, this is what I found out. I found out that I have a child that is blood type O, which is recessive. And it's the predominant blood type in our population is type O, which is recessive. The way that type O is written in genetics is with a little I, because there's no information. So if I have a child that's blood type O, recessive, didn't I get to give him one of those chromosomes? So, okay, so now from having a child that's blood type O, guess what? I know my what? Genotype. I know my genotype. My genotype is, I'm type A, but I'm heterozygous, right? I got one set of information from one of my parents. I don't know which one. And I got a different set from another. You with me? All right. And did I find out something about Scott? I still didn't care. Okay. So, okay. But I found out he's at least got that, right? Okay. So, but guess what? Then I had another child who gave blood, and when they gave blood, they got their little card from the Red Cross or American Heart. And that child is type A, B. This child is type O, which means kind of zero, no antibody, no antigens. And this child is what you call AB, blood type. Who gave this child the A? Me. I gave this child the A, but who must have given her the B? Scott. So now did I know Scott's blood type? He's type B, right? I'm A, he's B. We had a child that was O, which meant that we're both holding different, right? But you can have a child that's A, B, and it means that they will express both this one and this one. So it's called codominance because they're both going to be expressed. They're different sets of information, but are they both going to be expressed? They're both going to be expressed. That's what codominance means. Are you good with that?
So where does the positive and negative come in?
Okay, that's that's called, yeah, so that's rhesus factor, and we're going to talk about that. That's actually a different type of inheritance. That's inherited like the other thing I talked about. You're either going to be dominant or you're going to be recessive. Recessive is being negative. Dominant is having the factor. But this ABO blood typing is inherited as a codominant type of information. Because if you get from one parent the A, you will produce it. But if you get from the other parent B, you will produce that too. Do you get what I'm saying? And they'll both be there. All right, so that's just that's what codominance means. Is there blending? No. No. All right, and then I want to let you all know, this talks about codominant, with the ABO blood typing. This is FYI. Don't really ask you about this. We'll take a break here in a minute. Okay. Y'all all right? Okay. So there's some genes that contribute to one type of phenotype. We talked about that. We said that skin tone. It's got a lot of alleles. Eye color, alcoholism, heart disease. So there are a lot of different genes that are contributing to what you're actually seeing happening. This is why, though, family histories, are they important if you've got them? They're really important because really, honestly, we're probably going to figure out that everything has this genetic component to it. And the way epigenetics play a role is really helping us to figure it out. So some things, and this is this polygenic. Poly means what? Multiple genes contributing to I don't ask you this, I just want you to not be scared of the words. Are there any such thing as big words? No, they're just parts of words and you just mix them up, play them, whatever. So pleiotropic means that there's a gene and that one gene is going to have many different kinds of effects. So an example of this that I told you guys is devastating is sickle cell disease. With modern medicine, though, can sicklers avoid crisis? With modern proactive medicine, sicklers can avoid crisis. What each crisis does damage that's lasting. So you want to avoid crisis, want to be really proactive, and they can live normal lifespans, and that's wonderful, right? Same thing I told you about, like cystic fibrosis. Now that is a much shortened lifespan, but it's like a trait that's inherited that ends up being devastating to multiple organ systems, right? Okay. Now, I need to talk to you about sex chromosomes. Do you want to take a break before it or after it? Before it? Okay, let's take a 10-minute break and come back. Okay, 10 minutes, we'll be back. All right, so guys, let's talk about that 23rd pair of chromosomes, okay? The ones we call sex chromosomes. You all are going to know that this pair of chromosomes, they're called that because there is one trait on them among many others that are located on this pair. But there's one that might be present if the Y chromosome is present, if this little Y, which is much shorter than the X, but if this little Y is present, if it is present, there is a region of it that actually will hold information that's called the sex region of the Y, the SRY region of the Y chromosome. So it's known as the SRY, sex region of the Y chromosome. So if that Y chromosome is there, that gene will be expressed. So I want you to think about a baby. Okay, so if this if this baby has a little Y chromosome, and we know it's an X form, but a little Y, it looks like an X. It looks like an X, doesn't it? But it's a little short, and it's called a Y chromosome. If the Y is there, then the Y will be expressed, and this baby as an embryo, this baby, that little Y, that gene will be expressed and turned on, and it means that this baby will be flooded for not very long, but for a few hours during embryonic development. This little SRY gene is turned on just for a few hours, and this little baby gets flooded with testosterone, a type of testosterone. And when this baby gets flooded with this type of testosterone, the structures that, if it wasn't there, would have become the labial folds in a girl, become a scrotal sac. The structures that would have become the clitoris become a penis. The structures that would have become the fallopian tubes, you're going to end up getting ductus deferens, vas deferens. So because of that short window of time that the SRY gene gets turned on in an embryo, this little baby is a boy. But in the absence of it, if there's no Y chromosome, that would mean there's no SRY gene. And you have two X's. And so in the absence, guess what you become, ladies? You become a girl. Is that right? Does everybody get that? So gender is determined by one of the traits that are found on this 23rd pair. If the Y is present, it will be a boy. If the Y is absent, and the mother gave an X and the father gave an X, if the Y is absent, this individual becomes a what? So I hate to kind of tell you this, ladies, but we became female by default. Really? So okay. Now look, we're talking about when things happen normally. This is what would happen. Are we good? All right, so that means that when we look at a karyotyping, now y'all think about this just being that 23rd pair on the karyotype. We know we had all the first through the 22nd too. Now let's just ignore the male here and let's pretend we have a female. Are we good? We have a female on the karyotyping. And you guys said that when a female makes an egg, she can either send this X or she can send this X. But that's all she's got. Ladies, our eggs can either get one of the chromosomes that's an X or they can get the other one. But that's all we've got. Where men, when they make sperm, they will send half. Their sperm can actually get this X, or half their sperm will get what? The Y. So which gender parent determines the offspring's gender? The males. So if you've ever heard a man say that, my wife just won't give me a son, well, what do you know? He doesn't know much about somebody said he's stupid on her. I said he's stupid. I did not say that. So no, it just means he didn't understand genetics. But yeah, it's his sperm that are dictating the gender. Because the man's sperm, if the X sperm gets there, that embryo will become what? A female. And if the Y sperm gets there, male. Are you with me? All right, so there are some things, we said it's not just gender though. We said that there's a ton of different other traits that are held on the X and Y chromosome. There are other traits that are involved there. There are traits that have to do with blood typing, not blood typing, with blood clotting, traits that have to do with color, if you're going to be able to see colors, traits that have to do with your mental acuity, your cognitive ability. So let me say this. If you're looking at this individual, which you say this individual is? This is a female. So if there are traits that are found on the X chromosome that are responsible for all those things I just told you, plus there's more. But if there are all those things, then how many chances does a female get at them? Well, this one came from there, and this one might have come from there. I mean, you know, whatever. We're going to pretend the first one came from the mom, because mom should be first. And the other one came from their what? So, right. So how many chances do women get at traits that are found on the X chromosome? They get 2 chances at it. Do you get what I'm saying? Where males, if it's a disorder that's found on the X chromosome, males only get how many shots at that? One. And where do males get the X chromosome from? Their mother. Do you understand that? Does this make you see why? And again, Mendel talked about this. Does this make you see why that sex-linked disorders are more often in men? Does this make you see why? Because men only get how many shots at that information? And because they got that X from their what? And they didn't get an X from their dad. Are you with me? So men are much more often the ones we see with sex-linked disorders, like multiple muscular dystrophy. If muscular dystrophy is a great example of this, because again, and I don't know if y'all know, but the typical, there's 9 different types of muscular dystrophy, but the typical muscular dystrophy, those males don't live past 18, 19. That's a devastating disease, isn't it? There's some variants of muscular dystrophy that can be found in females. It's usually lesser though, but the traditional, the most common one, is the one that, you know, it's kind of devastating. Where do males inherit their disorder from? Which parent? From what? Their mothers. Their ex, right? So if you are male with a sex-linked disorder, can you send it to your daughter? Yeah. You could, because if you had that disorder, it's on your X, and if you have a daughter, she's going to have that X. But as long as she got the regular information from her mother, she shouldn't have a disorder. Is that right? But what about if the mother sent this X and the father sent this X? If this mother sent this X and this father sent this X to their daughter, would this daughter have it? It's just not as common in females. You get that? Sweating like whatever sweats a lot. Okay, so yeah, is that everybody good? Can a male with a sex-linked disorder send it to his son? Why not?
So the son gets it from the husband's partner.
Can a male, this is a male with the sex-linked disorder, can he send it to his son? No, he cannot, because he's sending to his son what? And the Y doesn't have anything to do with that disorder. Do you get that? He can't send it. But if he made it with somebody who had it, the child might have it. But can the father send it? No. Because the father sends to his son the what? And that's not on the X. You're right. I mean, that's not. You know that. Okay.
So just to clarify, the sex-linked disorders are typically on the X chromosome.
Yes, sex-linked disorders are on the X chromosome. Sex-linked disorders are typically found in males because males only get one what?
X chromosome.
Where females will have 2 shots at it. Do you get what I'm saying? All right, so does everybody good with that? Are the 23rd pair of chromosomes just dealing with gender? No, there are a lot of other very important traits we have to understand come from those, that pair of chromosomes. Are we good? Okay. All right, great. And I had talked about I had told you dominants and recessives don't have anything to do with population. You know, I promised y'all I don't read the PowerPoints, y'all, but that's example. Maybe y'all wish I did. But anyway, all right, so any questions from you so far? No? All right. So I want you to know that I want you to know about cancers and some terminology, if you will. So tumors, neoplasms. This is going to be, when we think about tumors, the study of them is called oncology. Oncology. Not all tumors are malignant. Some tumors are benign. Does that mean they don't have a chance of killing you? No. Some benign tumors can be deadly. And I want you to know that even though they aren't malignant, they can be deadly because they might be compressing vital organs. Like if you have a benign tumor in your brain, could that be deadly? Yes, because your brain is really your command center. It's controlling all homeostasis, isn't it? For everything that's going on in the body. So yeah, so benign tumors can be deadly depending on where they are and if they are compressing vital organs. And they're in a place that you can't get to them to remove them. Benign tumors are encapsulated, so they're staying where they are, you know, so because they're encapsulated. But if they're growing, you know, at too fast of a pace, then this is something to be addressed. Now, malignant tumors are very different. And when you think about malignant tumors, this being unencapsulated, it means that it's not contained. So it means that the cells, because remember, we're just all our cells working, right, together. The cells can slough off. And when they do, can they move to a different location? Can they be moved through your bloodstream to a different location and set up house at this different location? They can. So let me say this about malignant, you know, tumors, what you call cancer, if you will. And usually this is when the word cancer is, it's usually just used for malignant tumors. If that unencapsulated thing would just stay where it is, it probably wouldn't cause any death. But they don't, and this is the problem. They end up going to other locations and then causing problems in multiple locations. This is called metastatic cancer. And I do want you all to know that metastasis is just the process of these cancer cells moving, metastasizing. Y'all have heard that? So metastatic cancer is referring to cancers that have moved past their origin. You know, no one would die from breast cancer if it just stayed in the breast, would they? No one would die. No one would die from colon cancer if it just stayed in the colon, right? Prostate, same thing, right? But malignancies, cancer, malignancies, the thing about them is that they will and can metastasize because they're what? And they're usually fast growing. I need for you all to know something and hear what I'm saying. And you don't have to believe what I'm saying ever. And I encourage you not to do your own research. But you know, there is this conspiracy theory out there that there's a cure for cancer and the government is just keeping us from getting the cure from cancer. The government, along with pharmaceutical companies, are keeping these cures from us. That is paranoia at its utmost. That is ridiculous. I don't know if y'all know Joe Biden, but he lost a son to brain cancer not long ago. Bush lost a little girl. So, you know, the government, the boogeymen are not keeping And by the way, do you think that there are all these countries are now all of a sudden agreeing to keep the cures away? We have countries that will never be friends. I don't care, as long as Homo sapiens are on the earth, they will never get along, okay? But for that to be true, it would mean that you believe that there are these superpowers on the earth that are all coordinating this. Because research isn't just happening in the United States, is it? No, actually, we're behind on research compared to other countries, which is a real shame. You guys, it is paranoia. It is ridiculous. Those people also don't understand that cancer is not one disease. Cancer in the breast isn't even just one disease. There are many different types of breast cancer, which is different than colon cancer, which is different than the way that blood cancers can happen and prostate cancer. It is not one, it is many diseases. And here's what those same people don't understand, that when I started my research, and I did research too at UVA, when I started my career as a young person, like y'all, every year we could fill pro-game arenas with the children that died from acute lymphocytic leukemia. Now there's a 90% cure rate. Do you hear that? With most of these cancers now, we've come a really long way with curing these cancers. And actually, many cancers can be cured now. The whole idea is catching them early before they have what? Yes. And also, depending on what kind of cancer you're dealing with. Because is there one such thing as one cancer? No, there's so many different kinds. And guys, to think that someone is withholding, I mean, it's just really boggles the mind that people could really feel that way. It's not true. Does anybody want to add that they think it's true and have some proof of that? It's true. I mean, I would encourage you to speak now. Or otherwise, go talk to people that you know who espouse this kind of nonsense, because it's nonsense. It truly is. Truly is. Now, don't get me going about pharmaceutical companies, because there's plenty to talk about of bad things, and also insurance companies. But I can promise you there's not a cure for cancer out there that's being withheld. It's not. It's complex, you know, and it's different. But people are being cured. And what's just happened since my career, looking at this, there's lots to be happy about. Are there a lot of things that we could do to maybe shorten the length of time between approved meds and getting to the public? Yeah, I think there's a whole bunch of stuff that can happen that way. But no one is trying to keep cures away from anybody. And I would really welcome somebody to tell me if you have a different opinion about that. Would you feel comfortable to talk about it if you did?
Nope.
Why not? Why not?
I don't feel that way, but no.
Why? Because of the way I presented it?
Just because there's a classroom full of people, and it kind of feels
We
a
are all
little
friends. Classroom full of people, and you feel what?
A little intimidated.
By me?
No, just the amount of people that are eyeballing you. You just pretty much said, no, it doesn't exist. They're crazy people.
I didn't say the word. I didn't say
No,
I'm
paraphrasing. No,
I did not say the C word. I did not. I did not say the C word.
No, it's just intimidating.
Well, okay, that means that I presented, I feel strongly about that I can back up what I've said. So, and I would really welcome people if, you know, I don't want you to just think you have to listen to what I'm saying. If you think that there is evidence, say it. I would appreciate it. Seriously.
To play devil's advocate,
Sure.
how would you feel about the insurance companies being against it? I mean, I talk to insurance companies every day, and they deny things.
Oh, yeah. Like I said, insurance companies make me crazy. Okay, so like I use the C word, but I use it about me, right? So insurance companies and pharmaceutical companies both,
Right.
because they are private. These are private businesses. So bottom lines matter to them, but some things make no sense. Like some things really don't make sense at all. Like insurance companies, I just would have to wonder who's leading some of them, because sometimes they make you do things that actually end up adding to the cost in the end. And that makes and being turned down for something, if you just had this done without some of the stuff they make you go through, it would end up being cheaper. You know what I mean? It would end up being cheaper. So I don't know. I know this sounds really ridiculous, but I'm going to put it out there. There's a show called New Amsterdam, and they are bringing up now. A lot of it is so ridiculous. A lot of the medical stuff is ridiculous, but they're bringing up issues like this, insurance companies and the waste, and they're bringing up some really important issues that are happening in medicine. Is anybody watching that show? Yes. You haven't watched? They're bringing, and you worked in medicine a long time, Jane, because you sit back there quiet. But they're bringing up really ethical, important issues that are happening between pharmaceutical and insurance companies. I'm not saying it's a perfect world here, but there is not some boogeyman out there withholding a cure for cancer. Is cancer? It's not. There's not going to be a cure for cancer. I don't want to jinx anybody. There could be two people standing up here that have absolutely almost identical demographics. They're the same age. They're the same gender. They came from the same socioeconomic background. They were exposed to the same environmental things. You see what I mean? So they're very close. They can have the identical same cancer, be diagnosed at the identical same stage, but treatments will act differently in their bodies. Do y'all get the complexity? There's nobody that has a magic bullet for this. I can promise you if there was, they'd be making money on it. They would. They would not be withholding it. There are no, conspiracy theories are, really. I didn't use the C word.
Want me to say it?
No.
Okay. That's where I was going, like maybe the insurance, again, advocate.
I think it's because of lobbyists. It's because of lobbyists for insurance companies, lobbyists. And now we have a lobbyist who had never been allowed to be controlling, like head of human health and resources in our country right now is a lobbyist for Libby Pharmaceutical. Let's talk about a fox in the hen house. The lobbyist for EPA that is controlling the health of our environment was the head of EPA is a lobbyist for the coal mining industry. A lobbyist for oil and gasoline is now the head of our interior. You guys pay attention. Pay attention to what's going on. Some things don't make sense, right? So those decisions do all end up affecting outcome. I just was reading, what was I reading? It was important, and I was going to cut it out. I think it was in the week, but this was so important. It was a doctor who has been practicing for 30 years. And he said that back when he was practicing, early practicing, they were told in one of their ethics classes or whatever, if they read 8 hours a day, 7 days a week, the newest research, it would take 8 years for those practices to get to the practitioners, right? Because it's so much happening. But now, instead of it shortening, it has actually gone to 10 years. And so there's just so much happening. There's so much being discovered. So what it's going to take is good leadership to pull it all together for communication so that, and this is a lot to do with artificial intelligence, but by pulling everybody's expertise and crunching everything together, then we're going to be able to shorten that distance from what we're doing in research to get to the patient drastically. And so I think, maybe not in the near future, but I think in the next decade, with artificial intelligence and being able to get all of the medical data combined and shared, it's going to shorten the distance. And so that's exciting, guys. That's so exciting. It's so exciting because this is one of the ways that we progress so greatly in medicine is by sharing information and data. But yeah, don't get me going about insurance and pharmaceuticals. See, y'all got me on a tangent. I'm going to come back in. Look, I'm coming back. Causes. So what are causes of cancers? We know genetics plays a role in this, but anyway, carcinogens in our environment, our environmental agents. So we know smoking is not good for us, right? Dipping, smoking. We know alcohol plays a role. Food preservatives, exposure to radiation, right? Particles. Viruses, being exposed to certain viruses. We know that cervical dysplasia, cervical cancer is caused from human papillomavirus, HPVs. So write that down, human papillomavirus. That's not up here, but you would want to write that down. Cervical cancer, which is one of the leading causes of cancer deaths in females, cervical cancer is caused from exposure to human papillomavirus, HPVs. There are more than 100 different serotypes of HPV. HPV viruses cause skin tumors called warts, but you can be exposed to HPVs and never have a wart, but it could still be in your tissues. Most people who are positive for HPV have never had a visible or known wart. So some of these HPV warts will only affect your hands. Have y'all ever seen people who just had like little warts on their hands? Some of them will just do your feet, right? What do they call? Plantar warts, right? They're from viruses, HPV viruses, but it's a different type. And then there are a dozen or more, it's about 15, that can cause genital and oral lesions, warts, that we know can set you up for mutational changes that lead to cancerous changes. We're thinking now, we know that they're the ones that cause cervical cancer, but we're thinking now that more than 70% of mouth and neck and oral cancers are from HPV exposure. Here's the good news. Here's the good news. The good news is that there are immunizations against this. And so the Centers for Disease Control is actually recommending that kids even in middle school, because when the sexual activity kind of starts, I know it might not have with you, but anyway, but usually, because guess what? Puberty happens and your mind blows up. You know, puberty happens and you get curious, and puberty happens and you try things. So whatever, that's normal, but whatever. So they're recommending at middle school this series of, I think, so 3 immunizations. So this is going to save thousands of lives just from preventing cervical cancer. It doesn't prevent all HPV outbreaks, but it does, the ones that are out there now, these immunizations, they protect against the ones that lead to, can lead to cervical cancer. So even if somebody's been tested positive for HPV before, they should still get the immunization, because you might not have been exposed yet to the ones that, the types that can cause cancer. So get the immunization anyway, right? Sexual activity requires mature responsibilities, right, to protect yourself and others. So anyway, we also know herpes simplex is another one. They don't type them anymore, so this is a little bit old in thinking this, because cold sores are from herpes simplex, supposedly one, but they can be found on the genitals, and genital herpes simplex 2 can be found orally. So herpes simplex, very, very common, right? And that can actually lead to some changes. Hepatitis B virus and hepatitis C virus, these are both viruses, they can lead to liver cancer. So hepatitis B and hepatitis C. There are many other viruses. Do y'all know that hepatitis means inflammation of the liver? Itis as a suffix means inflammation, and hepatocytes are liver cells. So hepatitis, inflammation of the liver. There are dozens of things that can cause hepatitis. Alcoholism can cause inflammation of the liver. Just a lot of different things. Autoimmune diseases can cause hepatitis, but there are a lot of viruses that can cause that can attack the liver too. And hepatitis B and C are 2 distinct ones that we know can lead to liver cancer, may lead to liver cancer. Not always, but may. So we also have, okay, I'm not going to do that. But anyway, so mutagens. Mutagens are going to be something that can lead to mutational changes. So things that can lead to mutational changes can start interfering with the life cycle of a cell. And if the life cycle has changed and is now, the cell is just stealing from its environment, growing, dividing, we call it what? Cancer. Yeah, exactly. So we actually have a lot of defenses against these. So we have built in, because you're thinking, so why not everybody have cancer? You know, we all have such crazy lifestyles. I used the C word again. You started it. So anyway, no, we do though. We eat a lot of food preservatives, don't we? You know, we do that. But anyway, and we're living longer. And here's one of the things about living longer. You get to live longer, but guess what is more likely to happen as our cells are divided? Cancer. The incidence of cancers go up, okay? That's a lot better alternative to having a 40-year lifespan, isn't it? Well, y'all might not think so yet. Wait till you get there. So you're being glad that it increased. So some of this is just normal. I think what's happening in medicine now, we have actually gone to a point, and y'all need to be part of this and advocates for this. We've gotten to a point we can keep people alive way longer than maybe even makes sense. Do you get what I'm saying? Some of you might be like, Jesus. But absolutely we can. We can. So something to think about. How many of you have seen that happen? Yeah. And if you haven't been in medicine long enough to see, but you will see. We do things to humans that we would never consider doing to our loved pets. Not even in a million years will we consider doing it to our loved pets. Just to keep, I think we have to maybe get a little bit more zen and realize that, you know, it's a part of life, and there's a time, you know, and then the celebration of that. And I'm including myself because I'm a lot closer to that than y'all are, you know, and I'm including myself on that. We have, I think, 7 states now that actually have made it legal for euthanasia. Now, I do think there needs to be regulation, you know, obviously, but we have that, you know, and it's something to be thinking about. And you all will be hearing a lot more about that. Okay, not in this class, it's a different class, but okay. We have something called tumor necrosis factor, and this is, I've told you about peroxisomes, but tumor necrosis factor is something that our cells produce that actually destroy tumors. So we have built-in mechanisms to destroy any sort of mutational changes that occur, and then also even cancer cells. You know, some of my students have gotten freaked out when I say this, but from the time that you were created and until your whole lifespan, every day you will have cancer cells happening. There will be cancer cells happening every day. And a person who never has cancer. But what do you have? You have built-in mechanisms that take them out. So here's the deal. Keep your immune system strong, right? Limit your exposure to environmental whatever, and mostly keep your immune system strong, because that's what helps with it. All right. Oncogens. These are going to be cancer genes. What are you talking about? We have genes that actually code for cancer in our DNA? Yes. Yes, we do. Now, whether they will be turned on or not is going to depend on environmental factors. But we definitely do. So we understand that we have this. And we also have, built in our DNA, tumor suppressor genes that are counteracting some of this. So this is, y'all already knew this because you knew that if you have somebody in your family that has certain types of cancer, because it's cancer one thing, no, it's a lot of different kinds. If they have a certain type, there's some of them, not all of them, but some of them you can go as family and be tested to see if you're carriers. Is that right? Doesn't mean you're going to definitely have it, but it means that you have a much higher probability of having it than someone who didn't have the oncogens related to that. Now, what are the effects? Sorry, the title's into the text, but what are the effects? Nobody's copying the PowerPoints verbatim, but the effects of malignancies is that they're stealing nutrients from your functioning cells. So your functioning cells are going to all be suffering. Is that right? And they're going to sometimes block passageways. So they can cause bleeding. They can cause all kinds of things that can happen. And so these are just some of the things. We talk about cancer being a wasting disease, and it truly is, because what ends up happening is your glycogen reserves are going to be gone quickly, and then you're going to burn fat. And when your fat reserves are gone, you're going to start burning proteins, which are muscles, for energy. And so this is why we think about cancer as a wasting type of disease. When you're burning fats and proteins for energy, your body is thrown out of homeostasis, pH balance, and then that's going to end up causing a lot of other systems to be thrown out of balance. And so hopefully this kind of makes sense to you. I think you guys know that you've seen people with end-stage cancers and that sort of thing. How do you feel? Y'all feel okay? All right. All right, so what I'd like to do is, which we do have some things that we need to do, is actually have a little lab right now, okay? So this will kind of give y'all a break. Because during lab, unless it's something like really described, doing like a test, lab quiz or something, you can have your phones out. And this little exercise I really want you to, because you might be using them Google some stuff. On this thing we're going to do, and by the way, you can be taking vouching breaks during this whole time or whatever. But in the next 30 minutes, I'm going to go now and do this for you. I'm going to do it in here. I'm going to give you colored pencils, or, I got a lot of them. I'm going to give you colored pencils and paper, and you are going to draw a cell, a typical human cell. Now, on that, you're not going to be evaluated on your artistic ability. Did you see my baby? So you're not going to be evaluated on that. You're going to be evaluated. You're going to be evaluated by your peers on if you had all the structures labeled. And I want you to use colors in doing them. And for your structures, they don't need to just be labeled underneath the name, not on the back side, because I don't think you do all that flipping. Underneath your name, you have to have brief, just a few words, what their function is. Okay, so I'm gonna go get that stuff for y'all. Y'all get up, your book, maybe put out a book, and we're gonna take about 20 minutes or so. Now I have another exercise we're gonna do too, but this one is gonna count when I think I'm gonna give y'all about 20, 30 minutes to do this. This is gonna count, and I'm gonna check off your grade as a lab quiz participation grade on psychology, okay? So, by the way, you're going to get to keep it because will you study it for your test? Yes. Will you have multiple choice questions about functions of organelles and structures in a cell on the test? So I call those gimme questions because you should know it by now, or you'll know it after you draw. Okay, I'm going to get the plant color next to you. Y'all can take it, you know the rest, right?
Wait. Oh, yes.
It didn't have a brief. I had to add that in. It said, it says there is a bracket in the house, but in the quote, that's why this is not. It says the back side will actually just provide the crumbs, and then said that medium is equal. So I've gotten it wrong. I was thinking, she would say it.
Yeah.
Well, I've had to buy the multiple times. You're gonna do the softball? How you get to feel it?
Yes.
It's gonna be fun.
Okay, so your table, so somebody will come up, y'all can take this paper. Send somebody one person from your table, maybe. Okay, can we have other answers? Remember, your cell is not going to be a real cell because your cell on the outside is going to have a flagellum. And really, we only know one cell that has a flagellum. What is it? Sperm. And on the outside of your cell, in another place, you'll show some microvilli, right? And then you'll also show cilia. You know there's no such thing as a human cell that has all three of those things, right? But you're just putting them so that you can be aware of what they would be really looking like on the cell, right?
Yeah.
The heart ache for a moment.
Oh, are there any extra papers? I can go get some more.
Oh, I have one.
Julie, here. No, it was over here.
Oh, okay.
Did we need any extra papers? Oh, you did. Oh, I'm going. I'm going to get them. Some more papers. Who raised their hands? Anybody else? So guys, none of the artistic abilities, you know, mostly just being complete, okay?
I'll dance over. I can read it.